If you're ready to start a gene delivery experiment and need to create a brand new vector, our vector design wizard is chock full of our optimized and validated backbones and is integrated with our database of hundreds of thousands of components, from promoters and ORFs to tags and linkers. Click here to enter the design wizard and start by selecting from a wide variety of model systems and applications. Whether I choose lentivirus, AAV, regular plasmid, or transposon, the backbone is preloaded with everything I need for my experiment. I just need to slot in the components for expression in my target cells. Today, I need a lentiviral vector to express IGF-1 in cultured muscle cells. In the design page, I have everything I need to create my experiment-ready vector, including access to resources like guides and citations. I have the option to add multiple ORFs to create a polycistronic vector, but today we'll keep things simple. I can paste the sequence for my promoter, but VectorBee's incorporation of VectorBuilder's expansive database of ubiquitous, tissue-specific, and inducible promoters allows me to add my promoter of choice in a single click. For the ORF, I can search through VectorBuilder's popular ORFs, or I can utilize the integration with NCBI to search for my gene of interest in virtually any model species. I'm looking for human IGF-1, and I have a number of options for transcripts of different lengths. I can learn more by clicking on the RefSeq ID to go to NCBI, or I can go with VectorBuilder's recommended transcript. I'll add it to my vector, and either before or after adding, I can make edits to my ORF easily. For instance, changing the sequence with track changes, checking the translation of my ORF, or adding a tag to either the N or C terminus. Finally, I'll add my marker. We have a list of popular markers here, and I'm looking for a dual reporter. Now, VectorBuilder's database is not just expansive, it's also smart. The combinations in these dual reporters have been validated in the specific vector systems. For lentivirus, VectorBuilder scientists have found that the combination of EGFP and puromycin resistance genes work best when designed with a 2A linker in between, while EGFP and neomycin resistance genes require fusion for optimal function. Today, I'll use EGFP Puro. My vector design is all set up, so I'll officially finish my design. But VectorBee is my friend here, because the Vector Design Wizard is equipped with expansive knowledge of vector design thanks to decades of collective cloning experience. The Design Wizard will catch common design issues, including vector designs that exceed carrying capacity, and here the incidence of repetitive sequences in lentiviral vectors, which commonly results in recombination. Because I use two CMV promoters, I risk losing the sequence in between them, so I'll go back to edit my vector and replace the CMV promoter driving my gene of interest. I'm going to replace my promoter just as easily as I picked it, and this time I'll choose EF1A. Now I can finish my design, and I'm taken to the vector information page for my custom vector. Here it has been assigned a unique vector ID, and I can see all the relevant information for the different components, size, and growth conditions. If I'm all set and ready to have this vector in my hands, I can click Order from VectorBuilder, which will take me to the VectorBuilder website where I can quickly add this to my cart, along with any downstream services like plasmid preparation, virus packaging, and stable cell line engineering. Alternatively, I can direct this vector over to the project dashboard, where I can make further annotations, perform analyses, and, if needed, make additional changes to my vector. VectorBee's Design Wizard is a fast, user-friendly addition to the software, allowing users to create their ideal custom vectors in a few clicks. But because it's VectorBee, you still have the additional limitless customization, annotation, and analysis tools at your fingertips. So whether you want to move from idea to experiment as quickly as possible, or you want to spend time getting to know each nucleotide, VectorBee has you covered.